As a reminder, resting membrane potential. So at rest, there's a higher concentration of potassium inside the cell, and then a higher concentration then of sodium that is outside the cell. Okay, and so the opposite is true then. For potassium, we have a lower concentration outside the cell and then a lower concentration of sodium inside the cell. Okay, so at any moment in time, this is true for any place within the axon. So inside, this just represents the axon is more negative, and so it is going to be at negative 70 millivolts, whereas it's going to be more positive on the outside of that axon. Okay. All right, so we need to get into some terminology where we look at different electrical states of the membrane. So the first is, what is polarization? So at rest, a neuron is considered to be polarized because it has a more negative charge as compared to its outside. And so this is at negative 70 millivolts. Depolarization now occurs when the membrane is less polarized, so it becomes more positive than at rest. So this could be anywhere from negative 69 all the way to like positive 100. Okay, so it's become less polarized, so it's becoming more positive as compared to rest. Repolarization now means that the membrane potential is going back to that of negative 70 millivolts, okay, back to the resting potential. So typically, this occurs after depolarization has occurred, and so the values are becoming more negative. Hyperpolarization, hyper means more, and so this is when the membrane is more negative than at rest. So this is going to be like negative 71 millivolts, and anywhere, I mean, it could go to negative 1000 millivolts, for instance, or anything further, to be honest. So let's look at the different changes in the membrane potential. Okay, so first, when we have an upward deflection, this means a decrease in potential, and then a downward deflection means an increase in potential. So if we look at the y-axis first, what we have here is our membrane potential in millivolts. So at negative 70, here we're at rest. All right, I'm going to change my color too, so we don't get confused. All right, so here at stage 1, we have an upward deflection. So this then is becoming a depolarized here, okay, because we're becoming less negative. And so you can see the difference then when we look at the voltage, it is at around negative 58. All right, then at stage 2, right here, we have gone now downwards, and so we have increase in potential. And so now we're back to negative 70. So at this stage, what we would be at for number 2 is repolarized, okay? Because we went from a more positive to a more negative phase. All right, then at stage 3, here, we have an even further negative deflection or downward deflection. So we are continuing an increase in potential. So if we look at the voltage now, here we're about at negative 80. So at stage 3, we have become hyperpolarized. And then when we go back to normal, here at stage 4, we see the voltage is again back at negative 70. And so now we are maintaining our resting membrane potential in terms of this graph. So ion channels will affect the membrane potential And so this will be dependent on different ions. So let's first look at potassium, which again, potassium is going to be the predominant ion that will affect the permeability and potential of that cell for that neuron. So what we have here is that potassium channels tend to be inhibitory because they have a reversal potential of negative 90 millivolts. And reversal potentials are for an ion in the membrane potential at which there's no more net flow of that ion from one side of the membrane to the other. So in other words, this is at equilibrium for that ion. So for sodium then, this tends to be an excitatory ion channel, and that's because its reverse potential is at positive 60 millivolts. Chloride ion channels tend to be inhibitory because their membrane potential at equilibrium is between 60 and 70 millivolts, whereas calcium is excitatory because it has a reversal potential at positive 50 millivolts. So the question is, first of all, do you remember what was the resting membrane potential? So it was negative 70 millivolts. So anything that's excitatory, such as sodium and calcium, this is going to cause then depolarization, because we'll have movement with that ion generating a less negative response versus potassium and chloride, which those will have a either hyperpolarized state, such as especially with the negative 90, and then with chloride, what we'll have is a repolarization, because that's closer to the resting potential of negative 70. Ions within the axons are also gated. And so we'll talk first about the type of potassium channels, and then we'll talk about the type of sodium channels. So potassium has two types of channels. One that are gated, or not gated, sorry, these are always open, and so these are termed our potassium leak channels. So that means that there's always a constant leakage of potassium out of the cell, again, because there's a higher concentration of potassium inside the cell versus outside. The second type of channel that is for potassium are voltage-gated potassium channels. So when we hit a certain membrane potential or certain voltage, this will either close or open that gate, and that will either allow or prevent potassium from coming in or out. And so in this diagram here below, what we have is at a voltage of negative 70 millivolts at rest, this voltage-gated channel is closed. However, when we have a voltage that's more depolarized at negative 50, this will cause that voltage-gated channel then to open. Then when we go back to negative 70, then that channel will close again. Now sodium is a little bit different because it only has voltage-gated channels that are closed at rest, and then the membrane is less permeable to sodium at rest than it is to potassium. And a lot of that also has to stem from those leaked channels of potassium. Now, as we continue talking about these voltage-gated channels, these are important for action potentials because if you think about it, both potassium and sodium have a charge associated with them, okay? And so that means that these charges are either entering or exiting the cell. And so that's what's generating that electrical activity for that axon and for that neuron. All right, so if we continue on with voltage-gated sodium channels, so these channels open if the membrane depolarizes at -55. So in this figure here, -55 represents a threshold limit, okay? And so sodium then at that point is going to rush into the cell. And again, it's because now we have those channels open. And sodium is, by means of diffusion, will enter the cell going from high concentrations outside the cell to low concentrations inside the cell. So the membrane is going to be flooded with these sodium molecules, and that's going to keep increasing or depolarizing that cell. These channels, though, become deactivated at positive 30 millivolts. So they're going to shut down and not allow for any more sodium to come in. Okay? And so now let's see what happens next once we have those channels that are closed for sodium. So here we ended the last slide at positive 30 millivolts. So again, sodium had come in because we hit our threshold at -55 as compared to rest, which was at -70. Okay, so then what happens is that voltage-gated potassium channels then are going to open when we hit this -30. All right, so I'm going to change color so we see green now represents potassium. Okay, so because the membrane potential for sodium or potassium was at -90, this means it's going to now repolarize the cell because potassium now is going to rush out of the cell, and that follows its concentration gradient because we have higher concentrations of potassium inside the cell versus outside the cell. So by diffusion, it wants to move out. And so this will continue on until we get to about -80, -85, where we become hyperpolarized. And then over time, with the sodium-potassium ATPase pump, then we'll go back to our resting membrane potential. For an action potential to occur within a neuron, we have this all or none law. And so what happens here is that once we hit threshold, which again, threshold was at -55 millivolts, and that's when the voltage-gated sodium channels open and sodium rushes in. That causes depolarization, causing more sodium channels to open and more sodium to come in. And so this is an example, actually, of positive feedback. So this rapid depolarization is called an overshoot of that membrane potential until it reaches that positive 30 millivolts, when then the voltage-gated sodium channels become deactivated, and then we'll start the activation of the potassium or voltage-gated potassium channels. Now, if it gets to a point, though, where the threshold is not met at that -55, that means an action potential will not happen. So the -55, when we hit threshold, we have to hit threshold in order for an action potential to occur. So no matter the size of the stimulus, it will not affect the duration of that action potential either. When we hit that positive 30 millivolt mark, those voltage-gated sodium channels close, and again, no more sodium will enter, but now voltage-gated potassium channels will open, And then this is going to lead to potassium leaving the cell and causing repolarization of that membrane. So again, due to the fact that the reverse potential of potassium was about -90, we see this overshoot that is in a hyperpolarized state. And then at this stage, now the voltage-gated potassium channels are inactivated, so no more potassium can leave the cell. And then the sodium-potassium ATPase pump is what's responsible for reestablishing the resting potential for that neuron. And so this phase here that we have would be an example then of negative feedback. So here's another visual to look at this in terms of sodium and then for potassium. So with sodium, we have a net increase in ions moving in, and then that's caused by potassium to move out of the axon. Here, when we look at diffusion of the ions, so we have at this stage of sodium channels opening, then the inactivation, then we'll have gated potassium channels that are opening, thus deactivating that, and then inactivation of those potassium channels, and then no more movement of ions when all the channels are closed. So to sum up with the action potential then, when we have some type of triggering event, so this could be in the form of ions that have moved across, predominantly with potassium, that's going to cause a depolarization, so a decrease in membrane potential, and then it's going to open voltage-gated channels and to continue increasing those voltage or increase that sodium into the cell. And that will continue though until those channels are going to be inactivated, where then we start going back into negative feedback, where now potassium and the potassium channels are going to reestablish that membrane potential to rest. So we have a mechanism called refractory periods that are important because these occur after an action potential where the neuron cannot become excited again. So you can't overexcite neurons, which would be terrible because we would probably not be able to sleep ever. And so there's two different phases of refractory periods. The first is an absolute refractory period, and so that's denoted here in this figure. And this is when sodium channels have become inactive, where no matter how much of a stimulus you induce for them, you can't generate another action potential to meet threshold. A relative refractory period, in the blue here, this is the period in which potassium channels are still open, and only a very strong stimulus can overcome this. So it has to be something that's even larger and greater than what was before. So like in terms of memorizing new material, it's not something that you are familiar with, rather you like memorize or can learn something that you haven't learned before. Okay. So the big thing that we can change then is the frequency of action potentials, but you can't change how great the response is from them. So when we look at this visual, it helps to show too that we have propagations of action potential that travel down the axon rather than being sent back to the cell body due to these refractory periods, okay? And so that means that there's no backward current because of refractory periods, and so we have this forward current of action potentials that will keep going down towards the axon terminal. Again, as we said earlier, is that we can't generate a greater action potential. Rather, we can only affect the frequency of the action potential. And this is what's termed the coding for stimulus intensity. So strong stimulus may make action potentials occur more frequently, so like what we see here in this figure. However, a strong stimulus can also activate more neurons in a nerve, and so this can cause more recruitment. Since action potentials are all or none events, a strong stimulus, though, can't produce an action potential of a greater amplitude. So, rather, identical action potentials are going to be produced more frequently. So factors that influence conduction of action potentials down an axon includes the diameter of the axon and myelination. And so this will help reduce resistance to the spread of the charges. Thus, when we have a myelinated axon that's say 44 or 40 millimeters in diameter, that's going to conduct the action potential a lot faster than say a 10 millimeter diameter axon that's unmyelinated. There are two types of action potentials that can be propagated. The first is contiguous conduction and the second is saltatory conduction. So with contiguous conduction, what we have here is that the action potential will be conducted primarily in unmyelinated fibers, so typically within those small diameter axons and the very short ones. So action potentials are produced down the entire length of that axon, okay, and at every patch of the membrane. So you can imagine if this was for a large axon, this would take a lot of time. And so, for instance, if you stepped on a nail and we didn't have myelinated axons, then it would take you maybe several seconds to respond versus several milliseconds to respond. And so this conduction rate then with contiguous conduction is slow because so many action potentials are generated at each individual event. So here is an example of the slow conduction. So the amplitude of each action potential is going to be the same, and so it will continue to be conducted without a decrease in the response. Saltatory conductions, on the other hand, are going to be for rapid conduction in myelinated fibers, and these impulses are going to jump at those nodes of Ranvier. And so the jumping is due to the high internal resistance and the spread of charges within that membrane. And so myelin, again, is what provides the insulation, improving the speed of cable properties. So the nodes of Ranvier then have high concentrations of sodium channels at the nodes. And that allows sodium and potassium to cross the membrane every 1 to 2 millimeters instead of the entire duration of that axon. And so this will lead to faster conduction of action potentials and do not have to regenerate at every single section of the axon.